Amino acids are considered to be vital in our body. It is considered to be the building blocks of proteins, which are very essential for the structure and support of our cells. It is also used in building our muscles and repairing body tissues. This study used different virtual laboratories like Amrita, McGraw, and OLabs to gain knowledge and to explore the different theories of each test. In determining which amino acid is present in an unknown sample, there are numerous of tests available and each test reacts differently. For example, Millian's test gives a positive result if tyrosine is present in the sample, or the Hopkins gold test wherein a purple ring appears if phenyl is present. With that, this study aims to highlight the differences between the tests and the different positive results of each. Lastly is to gain a deeper understanding on each of the tests and their different uses.
Proteins or polypeptides are essential biological molecules that are composed of alpha amino acids and serve many functions. Some examples are the transport proteins, structural proteins, storage proteins, and many others. Proteins are complex organic compounds that contain nitrogen, carbon, oxygen, hydrogen, phosphorus, and sulfur. They are the polymers of amino acids that are joined by peptide bonds and chains. Amino acids are what make up proteins, and they're molecules that contain an amino and carboxyl functional group. They are mostly classified as zwitterions due to the isoelectric points having both positive and negative charges. Side chains in specific amino acids exhibit color reactions in testing, which can be used as a basis to distinguish them. The tests aim to be able to correctly identify the amino acid through the observed reactions and help understand the amino acid's chemical nature. These tests include the following: Buret Test, Pauli Diazo Test, Hydrant Test, Xanthoprotoic Test, Hopkins Test, Aguchi Test, lead sulfide test.
There were a total of 3 virtual experiments in Module 1B, 2 from Amrita Labs and 1 from McGraw. We first start off with part 1, which is called Qualitative Analysis of Amino Acids. This virtual lab had a total of 6 tests that would be used in a sequential way in order for us to identify which amino acid is present within the unknown solution. In this virtual experiment, we used beakers, test tubes, a test tube rack, a test tube holder, some droppers in its bottles, a Bunsen burner, an ice bucket, and a water bath. There were also various chemicals that were used in this experiment, such as, of course, our beginning product, which is the unknown amino acid, ninhydrin, the Milan's reagent, concentrated nitric acid, sodium hydroxide, sulfonilic acid, sodium nitrite, sodium carbonate, sulfuric acid, acetic acid glyxylic acid, and lead acetate. We start off with our unknown solution, which will be subjected to an Inhibition Test. This is done by adding 1 milliliter of the unknown solution to a test tube, followed by adding a few drops of the ninhydrin reagent. Then we shall mix the contents. After that, we place the test tube over a boiling water bath at 100 degrees Celsius for 5 minutes. And after that, we let it cool down to room temperature. This test may result in 2 different colors. A yellow solution would indicate the presence of the amino acid proline. But if we end up with a solution that is either violet, purple, or somewhat blue in color, this would indicate that the solution contains an alpha amino acid. Hence, we subject it to another test, which is the Xanthoprotoic acid test. In this test, we start off by adding 1 milliliter of the unknown amino acid to a new clean test tube. We then add a few drops of nitric acid, then mix it. Once mixing is done, we then heat the test tube over a Bunsen burner for a few minutes. And after that, we let the test tube cool by placing it under running tap water, or similar to the virtual lab wherein we placed it in a beaker containing room temperature water. The solution has now become yellow, but it does not indicate the end of the test. Once it has cooled down, we then add a few drops of alkali into the test tube. This would result into 2 different outcomes. An orange solution indicates that there is an aromatic amino acid present within the solution. However, if no change can be observed after the addition of the alkali, or if it retains the yellow color, This means that the solution has no aromatic amino acid within the solution. Moving on, these two results do not indicate a definitive end product. Instead, they will be subjected to two different tests. The yellow solution will be subjected to a lead sulfide test. This is done by the addition of 1 milliliter of the unknown amino acid solution into a new clean test tube. We then add a few drops of the 40% sodium hydroxide and boil it for 5 to 10 minutes using a Bunsen burner. We then cool down the contents of the solution and then add a few drops of the 10% lead acetate solution. This would result in a solution that has a black precipitate, which indicates the presence of the amino acid cysteine. Now, going back to the orange solution from the Centroproteic acid test, we subject it to a new different test, which is the Pauli's Diazo test. You start this off by chilling 1 milliliter of sulfonylic acid reagent in a small ice bucket. It is then followed by the addition of a few drops of pre-chilled sodium nitrite solution, and then we mix this. Immediately after that, we then add a few drops of the unknown amino acid solution that also has been pre-chilled, and once again we mix it. Lastly, we would then add sodium carbonate into the test tube in a dropwise manner until color appears. A red solution after doing this test may indicate the presence of tyrosine or histidine. Hence, we must subject it to another test, which is the Milan's test. But if there is no change in color or the solution remains clear, this may indicate the presence of tryptophan, which means we shall subject the solution to a Hopkins skull test. For the Milan test, we do this by adding 1 ml of the unknown solution to a new clean test tube, followed by the addition of the Milan's reagent, and then we mix it. We then boil the solution over a Bunsen burner for 3 to 5 minutes and cool the test tube under running water. Once the solution has been cooled down, we then add a few drops of sodium nitrite solution. This would result in a red solution, which indicates the presence of the amino acid tyrosine. Now, going back to the clear solution that will be subjected to Hopkins skull test. This is done by mixing 1 milliliter of the known amino acid solution with 1 milliliter of acetic acid glyoxylic acid. We then carefully add concentrated sulfuric acid to the side of the test tube in an inclined position. Once a purple-violet ring has formed, we can indeed confirm that tryptophan is present in the unknown solution. The second lab simulation is also done via Buret labs. The only difference is that it is no longer done in a sequential arrangement. Rather, it is done in order to identify the proteins or amino acids present in egg albumin and gelatin dispersion for comparison. Similar materials were used from the previous experiment. However, there is no longer the need for a water bath and an ice bucket. The chemicals used were egg albumin, gelatin dispersion, sodium hydroxide solution, 1% copper sulfate solution, concentrated nitric acid, and Milan's reagent. Of course, we start off by preparing the solution, but since it's a virtual lab, it has already been prepared for us. The first test we do is the Buret test, which starts off with the addition of the sodium hydroxide solution to both the egg albumin or test tube A and gelatin dispersion or test tube B. After that, we then add 1% copper sulfate solution to both test tube A and test tube B. After this, we then proceed to heating the test tubes with the use of a Bunsen burner for 5 minutes. And once a change has been observed, we then proceed to the documentation and record the results that we have gathered. For the next test, which is the Xanthoprotoic acid test, it is conducted by the addition of concentrated nitric acid in both test tube A and test tube B. We then proceed to heat both test tubes separately with the use of the Bunsen burner. Then proceed to the documentation of the results after a change has occurred. The egg albumin in test tube A and gelatin dispersion in test tube B are then subjected to a different new test, which is the Hydrant test. This is done by the addition of the Hydrant solution to both test tubes. It is then heated up with the use of a Bunsen burner, and once a change has been observed, the recording of the results follows. Lastly, in the Milan's test, which is done via the addition of the Milan's reagent to both test tubes A and B, when Milan's reagent has been added to test tube A, a noticeable change is noticed and hence should be recorded. After that, both test tubes are then heated up using the Bunsen burner, and then any observable changes may be recorded. The final lab simulation is from McGraw and is called Chemical Composition of Cells, Tests for Proteins. This test utilizes the Buret reagent in order for us to determine the concentration of proteins present within a solution. This test made use of materials such as graduated cylinders, test tubes, a test tube rack, steering rods, droppers in its bottles, and a wash bin for used slab materials. The chemicals used in the slab simulation are pepsin, albumin, starch solution, and the Buret reagent. Additionally, distilled water was also utilized in this experiment. We start off by preparing a test tube rack with 4 test tubes for each solution that we are about to test. We then add 2 milliliters of each solution to the respective test tubes, with the use of a graduated cylinder for precise measurements. We then proceed to label them in order for us to remember which test tube contains which solution. The 4th test tube shall contain distilled water, which shall serve as a negative control. Once preparation is complete, we then add a few drops of Buret reagent, and with the use of a clean stirring rod for each test tube, we use it to mix the solution with the reagent. Color changes can now be observed, hence it shall be recorded. And once the experiment is done, we identify the concentration of proteins of the solutions based on our recorded results. Lastly, in an actual laboratory setting, we must remember to be careful when doing such experiments and be properly clothed, and to take it slow, because there are no reset buttons that can magically clean your workspace and keep you from harm.
For the results and discussion, we have the following. So for the part 1 of the qualitative analysis of amino acid, we were able to utilize the software Amrita and the chemical test that we used, the Hydrant, Centroproteic, Pauli Diazo, Milan, Hopkins, Aguchi, and of course the lead sulfide test. The key instruments utilized in this simulation are the dropper, test tubes, water bath, and the Bunsen burner. So for the discussion, we were able to identify a possible sources of error, and this error is the instrument of choice. So the main instrument that was used to transfer reagents and liquids was through the rubber pipette, and this rubber pipette has a poor level of precision. So this could be a possible source of error. So that's for the part 1 of the qualitative analysis of amino acid. For the part 2 of the qualitative analysis of amino acid, it has a similar interface with the part 1. It is also under Amrita through OLABs. The chemical tests that we were able to use here are the Buret, Centroproteic, Hydrant, and Milan. It has a similar instruments, and it also has similar sources of error as the part 1, which is the instrument of choice. So that's for the part 2 of the qualitative analysis of amino acid. So for the part 3 of our simulation, we were able to identify some trends and observations in the macro hill virtual experiment. So for the tube 1, the pepsin containing solution, it resulted into purple, while the albumin containing solution turned into pink, while the starch and the distilled water resulted within the shade of blue. So for the observations, we were able to identify that the darkness of hue is directly proportional for the concentration. So with this in mind, we can assume that the pepsin containing solution, which resulted into purple hue, has the highest concentration. A change in color, typically within the variation of purple and pink, indicates a positive result and presence of protein. However, result within the any shade of blue indicates the absence of proteins. So within this in mind, we can say that test tube 1 and 2, the pepsin and albumin containing solution, has a positive result and has a protein present within the test tube, while the test tube 3 and 4 has no proteins in it. And let us not forget that the test tube 4 here has served as our negative control, the distilled containing solution. The key instruments utilized here are the graduated cylinder, the dropper, and the test tube. So we can see here an image beside me is an actual image of a pepsin containing solution and a negative control. I was able to retrieve it through high media laboratories in India. Here is a summary for the results of the second simulation, the OLAB simulation, which utilized an egg albumin dispersion and a gelatin dispersion. So for the Buret test, both of them resulted into a bluish violet. For the Hydrant test, both of the samples resulted into an intense blue. While for the Centroproteic test naman, the two samples resulted into a yellow precipitate. For the Milan's test, the egg albumin dispersion, it first was able to form a white precipitate. Then once it was boiled, it was able to form a brick red hue. So for the gelatin dispersion of the Milan's test, no changes was observed. In the image above me, we can see here an actual image of a protein egg albumin. I was able to retrieve this through Ain Shams University.
In this slide, you can see here a compilation of results for the chemical test. So for the Buret test, which is used for the detection of peptide bonds, the principle behind this chemical test is through the reagent reacting first to the peptide bond. This reaction yields a violet-purple color. For the Hydrant test naman, which is used for the detection of alpha amino acids, the amino acid reacts with the hydrant reagent. A positive result will yield into a violet-purple color, or also known as the Renaud's purple. For the Xanthoprotoic test, which is used for the presence of aromatic amino acids, the aromatic amino acids first undergo nitration. If nitric acid is present in the solution, it will yield a nitro derivative yellow color. The addition of NaOH naman will yield into orange due to ionization. For the Hopkins test, which is used to identify tryptophan, the tryptophan indole condenses first with the aldehyde. The condensation will yield into a violet-purple color. For the Aguchi Test, which is used to identify arginine, alpha-naphtol reacts first with guanidine. The positive result will yield into a red color. For the lead sulfide test, para ginagamit siya sa pag-identify ng mga sulfur containing amino acid like cysteine, the boiling of sulfur and containing amino acids together with sodium hydroxide will yield into a brown or black precipitate. This is a positive result. Lastly, for the Milan test, which is used for the identification of tyrosine, the tyrosine reacts with the mercuric ion. In acidic conditions, the presence of sodium nitrate will yield into a yellow-red, into a red precipitate. So that's for the compilation of the chemical test with the qualitative analysis of those.
So now we will be discussing conclusion and recommendation. So our group uses 7 different tests on protein. The first test is the Buret Test, which is used to detect peptide bonds. So the principle behind it is that the copper sulfate reacts with the peptide bond, which gives a color of purple or violet. The next test is the Hydrant test, which is used to detect alpha amino group. So for this one, the Hydrant reacts with the amino group, then it oxidizes the compound. After that, it releases ammonia, and this ammonia further reacts with the Hydrant, which gives a color of purple or violet, which we can also call Renaud's purple. The next test would be the Centroproteic test, which is used to detect aromatic amino acid. So for this one, the aromatic amino acid undergoes titration through nitric acid, and with an addition of NaOH, it began to ionize and it gives a yellow color. The next test is the Hopkins test, which detects tryptophan. The glycylic acid, which is the reagent used, combines with the 2 molecules of tryptophan acting on a indole ring. Then the condensation product form undergoes dehydration to form a violet-colored pigment. The next test is the Aguchi test, which detects arginine. So the alpha-naphtol and the guanidinium group in arginine reacts together in the presence of an oxidizing agent, the sodium hydrophomite, to produce a red color. The next test is the lead sulfide test, which detects sulfur containing amino acid such as cysteine or cysteine. So for this one, the SH and SS group are degraded through a strong alkaline condition. Then after this degradation, it produced a sulfur, which then combines with the sodium hydroxide, and its product reacts to lead acetate that resulted in a brown or black precipitate. The last test is the Milan's test, which detects tyrosine. So for this one, the phenol group of tyrosine is titrated, then it combines with mercury ions, and it forms a red color precipitate. We also recommend to implement the experiment in an actual laboratory because it will provide factual results and help researchers to determine errors that occurred in the experimentation.